you very much. So my area of research is in misclassification, measurement error, and I have a good friend that works for a pharmaceutical company, and he called me and they had a misclassification problem for both the efficacy and the safety. They were the test they were using was potentially going to mislabel, and so he was curious, would that impact the power? Okay, yeah, the power and the operating characteristics of their results. So it made me think that this would be something interesting to a clinical audience. First, a few things about Bayesian sample size determination. It can be different from the frequentist or classical approach in that you don't require fixed values for unknown parameters. For instance, if you have a variance, if you have a normal distribution, and instead of plugging in a value to get a sample size, you could use a distribution instead. And so what this does is you use these prior distributions as weight functions to weight all possible data sets, average over the sample space, and then you can determine the power or the interval width if you're using confidence intervals or whatever. And so again, we have all these different approaches for the method. If you're interested, I'm just going to go ahead and pull all this up at once. If you're interested in Bayesian sample size determination, the classic paper, and it's very readable, is Wang and Gelfand from 2002. And what they argue for is you elicit two different types of prior distributions. They call them sampling and fitting priors. Since this time, the terminology design and analysis priors have become more used, but they mean the same thing. So the sampling or design priors is what a traditional approach would use, plugging in a particular value. So you, for instance, you think an effect size is going to be 2, so you would plug in 2. Well, Wang and Gelfand say, okay, instead of plugging in 2, let's elicit a prior. It could be as low as a half. It could be as high as 3. So let's elicit a distribution and use that at the design stage. Then the fitting or analysis priors are your traditional priors in a Bayesian analysis, and these can be non-informative. So if you want a diffuse, flat prior, let the data completely do the talking, that is absolutely fine. So suppose we have this quantity of interest tau, and our posterior is pi of tau given our data. Then you have this marginal density, which I know maybe some of you aren't as familiar with Bayesian inference as others. The marginal density is sometimes referred to as the prior predictive. So in other words, it's before you see any data, it would predict every possible data set and would say, okay, this data set has this % chance of happening, this data set has this % chance of happening, et cetera, et cetera. And so this prior predictive is based on those sampling priors or the design prior. And so that way you elicit these priors of what you think may happen or what a regulatory agency says must happen. You know, you need to have an effect of at least 70% or something like that. So you get these design priors, and then the marginal will then show you what data sets are most likely to happen in that instance. And then we can do our sample size determination. There's several different types of sample size determination. Several are interval-based. So in other words, and I'm not going to get into the overly technical here. I'll try to put this in English. So what this essentially says is, I'm going to fix an interval width, and I'm going to find the data set, or I'm sorry, I'm going to find the sample size that guarantees that interval width on average, and or that coverage, that coverage probability on average. And so you say I'm going to want to be able to have an interval of length 3, and I want a 95% coverage, so this is going to find it so that the interval for surely will be length 3, and on average, my coverage will be 95%. That's one approach. Then we can flip that to what's called the average length, and that does the exact opposite. That finds the sample size that I'm fixing my coverage, so it's for surely going to be 95%, and on average, my length would be some specified length. And then finally, there's power, which is what I'm actually focusing on. And even though this is in a Bayesian context, we can use alpha and beta in sort of the same way. So what this is saying is, I want to find the sample size so that on average, let's use the traditional values for alpha and beta. So alpha is type 1 error, 0.05. Beta is type 2 error, 0.2. So then power would be 0.8. This says, on average, I want the probability of seeing an effect, tau greater than 0, controlling type 1 error at 0.05 is at least 0.8, the power. Okay? And then, of course, you can flip that. You can do two-sided. There's various options there. So there's if you were testing less than 0 instead of greater than 0. Certainly a common criticism of the Bayesian approach is that it's computationally intensive, okay? So we can't just, in 2 seconds very often, find an answer. We have to use Markov chain Monte Carlo, which has various issues, but those are going away every year as our computational power increases exponentially. So that's really going away. So now, again, I'm going to try to stay a little higher than what my slides would indicate. I'll try to keep this as non-technical as possible. The idea here is we have a diagnostic test that's fallible. D is the truth, diseased or not. T is what we observe, positive or not. And each test we have has a sensitivity and a specificity. And if you don't account for the misclassification, if you just use your test as a surrogate for disease, then you're not estimating your true probability of disease, P, you're estimating this observed prevalence, pi, which is a function of the true prevalence, the sensitivity, and the specificity. And so clearly, this is what my friend was concerned about, that there might be, that by not accounting for the misclassification, we might be doing something wrong, okay? And as Bayesians, we need priors for all of these parameters, and we'll use beta priors, the beta distribution. I'll show it to you right here for those interested. But the point is, it's a distribution that goes from 0 to 1, like a probability is supposed to, and it's also a very flexible distribution, so it can have a range of shapes and can be useful in terms of eliciting probability values. Okay, so here's what our data looks like for this one diagnostic test. So you've got, say, 2 samples here. And so N11 is the number of positives for the first sample. N12 is the number of positives for the second. N01 is the number of negatives. N02 is the number of negatives for the second. And you can see the various cell probabilities there. And again, if you don't account for the misclassification, this would be the underlying distribution, but you're trying to get at the prevalence. Whoops, typo, sorry. These should be 2s, population 2. And same thing for the, this is where we're headed with this research, and that's with 2 diagnostic tests. And again, this would fill out a larger table. Okay. I think this should be interesting because this is an algorithm for just Bayesian sample size determination in general that I think would be I would like to see used more often. It's a computational method, and what you do is you select a range of sample sizes that either are what you would expect or what you can afford, and for each sample size, you would simulate a parameter value from the prior distribution, from those design priors, generate data, compute whatever interests is in your study. So, for instance, if it's a confidence interval or if it's a power, whatever, you would compute it, and then you're going to do that a whole bunch of times, and this is where it's really computationally intensive. You're going to do this a whole bunch of times, and that would get an overall average power or average interval for that particular sample size. You're going to do this for a range of sample sizes, and then you can fit a curve to find what sample size you need, or you can sort of center in on what the exact sample size you need and run it a whole bunch of times at that little area and get an exact sample size. So anyway, so this is just the general algorithm. For us, we considered a case, again, this was the situation my friend brought to me. He wanted to look at 4 different doses, and they thought the sensitivity was really accurate. So that's this beta 98.2 that has a mean of 0.98, so very, very high sensitivity. They were a little more concerned about the specificities, so they were more concerned about the false positives. And so that was a beta 60/40, so about a 60% probability of a false or 40% probability of a false positive, 60% of getting correctly classifying. And anyway, then they had these four doses. They thought 2 of them were going to be very low, 1 kind of low, and 1 not so low. And so then we looked at this, and if you ignore the misclassification, this is the power for testing 1 versus 2, 1 versus 3, 1 versus 4. You can see the power is actually quite high where it should be, 1 versus 3 and 1 versus 4. This is trying to control at 0.05. And so the interesting thing is that we're not really controlling the type 1 error very well. You can see that it gets up to about 10% here when we're supposed to be controlling at 0.05. But when we should be detecting a difference, it detects it quite well. But remember, 1 and 2 were the same. 1 and 2 were the same probability. 1 and 3 and then 1 and 4 should be rejected. So that's that. Now, when we accounted for the misclassification, so now again, 1 versus 2, it should be 0.05 or less, and it is. But our power is much, much lower for the 1 versus 3, 1 versus 4. So that's not good. I mean, especially for the 1 versus 3. So again, let me go back here. When we ignored the misclassification, 1 versus 3 had a low power of 40% and gets up to almost 70%. When we control for the misclassification, it never gets much above 20%. So that's not so good. So the question is controlling the type 1 error that worth it that much to us that we're going to lose that much power? Now, but here's what's going on. Here's why the power is so high when we ignore the misclassification. So this is the coverage of the various intervals. These should all be right around 95%. We were trying to capture these things 95% of the time, and you can see that we hardly ever capture them even 90% of the time, and it just gets worse and worse as the sample size gets bigger. But if we account for the misclassification, it turns out for these guys, the really low probabilities, P1 and P2, it's between 85 and 90%, but the others, P3 and P4, is very good, around 95%. So we again, we need to try to figure out how to handle these really, really low probability cases, but the higher, the more just sort of small but not super small probabilities are handled quite well. So, in conclusion, low response probabilities and a single diagnostic test, we do not help power at all. Power is quite low, unfortunately, but it does control type 1 error. However, accounting for the misclassification does improve the bias and the coverage that is completely missed in those cases where we don't correct for the misclassification. Where we're going with this is we're going to add a second and possibly even a third diagnostic test, which will greatly improve the power when we account for the misclassification. Is a little more expensive, and we want to see if we can get closer to the naive case there. Any questions?